Hello dear students, myself Dr. Niro Soni, and today we will discuss the chapter 5 Principle of Inheritance and Variation MCQ. It is most MP for the Gujarat Board 2023 exam. So kindly focus on Ncert Exemplar, every chapter MCQ. Question number 1. All genes located on the same chromosome. All genes located on same chromosome, say for example, there is two gene A and B on same location because all the genes present on a particular chromosome form a linkage group. One linkage group, that's why it is located on same chromosome. The number of linkage group of a species correspond to the total number of different chromosome of that species. It is not simply that the number of chromosome in a haploid set. Say for example, human male consist of 22 plus XY chromosome, but male female containing 22 plus XX chromosome. So here 24 linkage, 22 male, 22 female, total 24 linkage in a female, 22 pair autosome and 2X chromosome. That's why 22 plus X in female and 22 plus Y in male if male child birth, but female child birth then 22 plus X. That's why 22, 22, 44, 45, 46 respectively X and Y chromosome. So while option A, C and D are incorrect, that's why option B should be correct over here. Question number 2. Conditions of a karyotype of 2n plus minus 1, 2n plus or minus 2 are called as, this is known as aneuploidy, because aneuploidy involves the changes in chromosome number of addition or deletion. Either it will be additions or deletions of less than a whole set. In this case, organism gains or loss one or more chromosome but not a complete set. Polyploidy is defined as the addition of entire set of chromosome. This polyploidy can be a triploidy, this polyploidy can be a tetraploidy, and this polyploidy can be a pentaploidy, etc. So allopolyploidy means, allopolyploidy is the defined as the polyploidy in which chromosome sets a non-homologous chromosome. Non-homologous chromosome. In other words, we can say that the allopolyploids are derived from a stock which is heterozygous, in conditions, say for example, capital A, small a. Monosome is the process in which the chromosome is removed from diploid set of chromosome. That's why it's called 2n minus 1. Option A should be correct over here. Distance between the genes and percentage recombination shows it is a direct relationship, because crossing over, it is known as recombination. And whenever the recombination occurs, that's why it directly lead to change of varieties of species. That's why crossing over, or it is known as recombination, is the mutual exchange of corresponding segment, the adjacent parental and maternal chromatids of this synapse homologous chromosome, producing a new combination of genes. Crossing over separate genes away from each other, so the distance between the gene and percentage of recombination shows a direct relationship. Say for example, when genes are close together, that have high linkage but exhibit low recombination frequency because they cannot separate. Thus, the other options are wrong. It does not show a parallel or inverse relationship. That's why we directly choose the option number A, direct relationship. Question number 4. If a genetic disease is transferred from a phenotypically normal but carrier female to only some of the male progeny, the disease is, your answer is sex linked disorder, are the genetic disease which the defective gene are carried on the either X chromosome or Y chromosome. But most sex-linked conditions are recessive. This means that a person to have two chromosomes, X chromosome, both X chromosome in females, both copy of a gene, one of X chromosome and other in also X chromosome. But one is affected, that's why it is considered as a carrier, but it is a phenotypically normal. But in male, one having X chromosome, one having Y chromosome. Mostly sex-linked recessive, sex-linked is gene present on the X chromosome in male. A female with a mutation, one copy of a gene on the X chromosome is said to be a carrier for an X-linked condition. So for X-linked disorder, an unaffected carrier mother who has a mutation in a gene on the X chromosome can transmit either the X chromosome with this mutation or but a normal X chromosome to her children. Autosomal dominant inheritance refers to a pattern inheritance in a condition directly or indirectly due to a dominant faulty gene located on autosome. But autosomal recessive inheritance is a condition caused directly or indirectly due to a recessive faulty gene copy of an autosome. Sex-linked dominant is a rare trait disease. Sex-linked dominant caused by a single abnormal gene on the X chromosome. That's why option D should be correct over here. Question number 5. Very easy question. In a sickle cell anemia, glutamic acid replaced by valine. That's why glutamic acid coded by GAG and valine GUG at 6th position. So your answer is GUG. Person having genotype IA-IB would show the blood group as IAB. This is because of only a codominance. Codominance is the example of blood group. Otherwise, it is considered as a multiple allelism. Now, question number 7, very most ambiguous question in your board examination and NEET examination. ZZ and ZW type of a sex determination is seen in birds as the ZW case. ZZ and ZW case. ZW case, the male has ZZ, that's why it's called homomorphic sex chromosome. Peacock shows the ZZ and ZW sex determination type, but platypus sex determination type is XX and XY. Both male and female has 10 sex chromosome of the each. The male has XY, 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 and XY, and female has XX, 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 and XS. The snails has the sex determination is environmentally induced, while in cockroach it is of XX and X0 type. In this type, Y chromosome is completely lacking, but in this presence of unpaired X chromosome determine the masculine sex. So your answer is ZZZW in peacock. Question number 8. A cross between two tall plants resulted in offspring having few dwarf plant. What is the genotype of both parents? Where is the question? What is the correct option? So say for example, tall plant. So one is considered as a TT cross TT and few small plant. If tall plant considered as a homozygous condition, so neither you will get a dwarf because both having tall and tall. So either you should compare the TT cross TT. In heterozygous condition, it is a tall plant, but gene is a heterozygous in manner. That's why TT, 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 and TT. So tall plant having 3 and small plant having 1 ratio. So pure tall plant having 2, 1, pure tall plant having 1, hybrid plant is 2, and dwarf plant having 1. So on the basis of monohybrid cross, a cross involve only one trait. Mendel formulated the law of segregation. Capital T, small t, capital T, small t. Let use the Mendel cross tall and dwarf pea plant as an example. The F1 plant of genotype capital T, small t are self-pollinated, both tall capital T but with dwarf small t alleles. What is back cross? What is back cross? The cross between TT cross TT, this is called back cross, is resulting in the two homozygous and two heterozygous dominant alleles or gametes. The cross between capital T and small t, this is called test cross. This is called homozygous recessive. So your answer is What is test cross? TT cross TT. TT cross TT. It is known as back cross. It is cross with homozygous recessive. But what is back cross? TT cross TT gives back cross. This is called test cross. TT cross TT, this is called test cross, but TT cross TT cross back cross. So this is very most important question. Question number 9. In diabetic cross, if you get 9331, the ratio is denoted that the cross that involves the analysis of two independent traits is termed as a diabetic cross. The law of independent assessment was deduced from the Mendel's experiment with diabetic cross. Alleles of two genes segregatedly independently, but it can be explained as: suppose crosses are made between a pea plant with round and yellow seed, one with winkle and green seed. All F1 hybrid give yellow and round seeds since yellow color is dominant over the green and round shape is dominant over the wrinkle. So phenotypically ratio is 9:3:3:1. Your answer is the allele of two genes are segregated independently. Question number 10. Which of the following will not result in variation among the sibling? Linkage refer to a physical association of two genes on same chromosome. That's why this is called linkage. Because linkage will not result in a variation among siblings. Morgan carried out a several laboratory cross in Drosophila to study genes that were sex-linked. Morgan came to know that the genes were located on X chromosome, but also observed that when two genes in a dihybrid cross were situated on the same chromosome, the proposed parental gene combination were much higher than the non-parental type. It indicates that due to the physical association of the two genes, there will no variation among siblings. Independent assessment of gene means that the allele pair separate during the formation of gamete independently. It means that traits are transmitted to offspring independently of one to another. Crossing over is exchange of genetic material between the same homologous chromosome. It is one of the final phase of the genetic recombination, and mutation is sudden inheritable change in genetic material of an organism which transfer to next generation. That's why linkage is correct over here. Question number 11. Mendel's law in independent assessment hold good for genes situated on the non-homologous chromosome, because the law of independent assessment hold true as long as two different genes are on separate chromosome. When the gene are on separate chromosome, the two alleles of one gene capital A and small gene small a will segregate into a gamete independently of two gametes and other gene capital B small b. Equal number of four different types of gamete: capital A, capital B, small a, capital B, capital A, small b, and small a, small b. But if the two genes are on the same chromosome, they are linked and will segregate together during meiosis, producing a two kind of gametes. Homologous chromosomes are similar but not an identical. Each carries the same gene in same order, but alleles for each trait may not be the same. Extranuclear genetic elements are also called as plasmid, but shows the pattern of maternal inheritance. So option A should be right over here. Question number 12. Occasionally, a single gene may express more than one effect. The phenomena is called. This is called pleiotropy. Because multiple allelism means what? Multiple allelism is a series of three or more alternative allelic form of a gene, only two of which can exist in any normal diploid individual. Say for example, genes in a blood group in human. But what is mosaicism? Mosaicism describes the occurrence cell that differ in genetic components from other cells of the body. But what is polygyny? What is polygyny? Refer to a single characteristic that is controlled by more than two genes. It is known as multifunctional inheritance. So that's why pleiotropy is the correct answer, because it is in our NCERT. Question number 13. In certain taxon of insects, some have 17 chromosomes and other having 18 chromosomes. The 17 and 18 chromosome bearing organism are. This is called male and female respectively, because in certain insects such as cockroach and such as some roundworms, Y chromosome is missing, so that the male has only sex chromosome, and say for example X condition in the male X0. X0 means what? Absence of on sex chromosome. In the female is XX, that's the male showing 70 chromosome and female showing 18 chromosome. All other options are given wrong. Question number 14. The inheritance pattern of a gene over generation among humans is studied by the pedigree analysis. The character studied as a pedigree analysis is equivalent to. Pedigree analysis is equivalent. Pedigree is a family tree that diagrams that relationship among the parents and the past generation and the next generation and children across the several generations, which shows the inheritance pattern of a particular phenotypically character. So that's why it is a Mendelian trait. Because Mendelian inheritance in a human is difficult to study. Current understanding the Mendelian inheritance in humans is gained by the analysis of family pedigree analysis, resulting in mating between the mating between the parents that have already occurred. By analyzing of pedigree analysis, we may be able to predict how to trait is inherited. It is a visual tool for documenting and biological relationship between family and to determine the mode of inheritance like dominant and recessive of genetic disease. There are different types of pedigree analysis: autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. In your syllabus, autosomal recessive and X-linked recessive given here. Another different types of pedigree analysis like Y, Y chromosome pedigree analysis, cytoplasmic inheritance, Y inheritance, and cytoplasmic inheritance and mitochondrial inheritance. These three are another types of pedigree analysis, but it is not given in your NCERT. So polygenic trait are the another exception to Mendel's rule, which occurs when a trait is controlled by more than one gene. That means the each dominant allele adds to the expression of to the next dominant allele. Maternal trait means what? Inherited. These maternal traits are the inherited traits, but expressed from the maternal parents to the subsequently offspring. So your answer is option B. Question number 15. It is said to the Mendel proposed that the factor controlling of any character is discrete and independent. This proposition was based on the, because the law of segregation state that the factors or alleles of a pair segregate from each other during the gamete formation such that a gamete receive only one of the two factors. They do not show any blending. The phenotypic ratio of diabetic ratio is 9 raised to 3 raised to 3 raised to 1, but genotypic ratio is 2 raised to 2 raised to 4 raised to 1 raised to 2 raised to 1 raised to 2 raised to 1. So result in a diabetic cross where the two parents different on in two pairs of contrasting trait seed color and seed shape. Result that's rest options does not support the Mendel law of segregation. The observation of that offspring cross made between the contrasting that have blending. So finally, rapid review of the answer in your NCERT Exemplar chapter 5. Question 1 containing option B. Question 2 containing option A. Question 3 having right answer option A. 4 having D. 5th में D. 6th में B. 7 में आंसर आएगा D. 8 में आंसर आएगा B. 9th के अंदर आंसर आएगा D. 10th के अंदर आंसर आएगा सी, 11th के अंदर आंसर आएगा ए, 12th के अंदर आंसर आएगा सी, 13 के अंदर आंसर आएगा ए, 14 के अंदर आंसर आएगा बी एंड 15 के अंदर आंसर आएगा हमारा बी. Now let us move on 16 number question. Question number 16. Two genes A and B are linked in a dihybrid cross when two genes F1 is a heterozygous is crossed with, say for example, heterozygous or BB cross BB or AA cross A. Homozygous recessive parental type of AABB, what would be the ratio of offspring of next generation? Say for example, look at over here, that's why different types of gene RRYY cross RRYY. What is the genotypic ratio? Our gene is RY, RY, RY and RY. Here only only one gamete RY. Here 4 gametes are over here. So RY, RY. The one is called round yellow RRYY, RRYY. Third one is called R. Third one is called RRYY and fourth is RYY. This is 1:1:1:1. The other options are incorrect over here. Because when a progeny F1 is crossed with the homozygous recessive parent, it is called as test cross. This is question from test cross. So always test cross answer is 1:1:1:1. Question number 17. In the F2 generation of a Mendelian dihybrid cross, the number of phenotype and genotype are. Mendelian dihybrid cross, the ratio is 9 genotype and 4 phenotype. Round yellow, round green, wrinkle yellow, wrinkle green. Question number 18. Option D, phenotype 4 and genotype 9. Mother and father person with a O blood group that have a A and B blood group respectively. What would be the genotype of mother and father? When a cross is carried between heterozygous father of blood group B and heterozygous mother of blood group A. So IB, IO and IA, IO. So AB, A group, B group and O group. So all the 4 blood groups are controlled by the 3 allelic gene IA, IB and IO. Thus it shows the phenomena is multiple allism. Both IA and IB dominate over. But however, when together both dominant and show the phenomena of codominance forming the blood group AB, 6 genotypes are possible with combination of these 3 alleles. Thus the other options are wrong and incorrect. Option C should be right over here. Because IAIO it is heterozygous, IBIO it is heterozygous, that's why other options are wrong here, wrong over here, because the child with blood group O will have homozygous recessive alleles. Therefore, both the parents should be a heterozygous. Say for example, genotype father will be IA and mother will be IB. So this is your NEET examination question as well as March board examination question 2023. Kindly prepare it. Best of luck. All the best, students. Thank you for your support and share my channel with your friends. Thank you.